안녕하세요. My name is Kevin Chen. I'm the head of mRNA teams in ProBio. It's a really great chance to come here for this discussion. In the presentation, I will show some information about our mRNA platform capabilities and also some case studies. In that way, we can have more discussion in the QA part. So today, my topic will cover the mRNA-tLNP platform capability for the in vivo CAR-T application. So my talk will cover two perspectives. One is advanced and challenging for the in vivo CAR-T therapy, and the second one is our all-in-one CAR-T CDMO platform from ProBio. So CAR-T, thanks for Dr. King's introduction in the first presentation. So CAR-T is very powerful therapeutic method to treat blood cancer or other type of cancer. And traditionally, we use the ex vivo CAR-T therapy to treat some cancer disease. And here, the picture I present here shows a typical process for the CAR-T production. Okay. So on the picture on the left, this is the process for the ex vivo CAR-T. So basically, first collect the blood sample from the patients and isolate the T cell, and then genetically modify the T cells and generate expand the CAR-T cell number, and then after the QC check, and then infusion back to the patients. So for the in vivo CAR-T production process, it's much more simple. Basically, we generate the CAR-T samples either in the non-viral strategy like LNP or using the viral strategy like the LVV virus. So we can infusion these samples back to the patients and generate or express the CAR gene expression in the patient's blood. So typically there are two main strategies to deliver the CAR genes into the patients. The first one is a non-viral strategy. We use the lipid nanoparticle as a delivery format. And the second one is a viral strategy. We can mutate the lentivirus and also add some binders on the surface so that in that way the LV can specifically target in the tissues we want. And here is the table shows a comparison between these two methods. Either the integration is a transient expression or stable integrated into the genome. And the targeting efficiency is also a little bit different. The LNP will be moderate with off-target effect as a key challenge in the CMC process. And the lentiviral has a higher targeting efficiency because it's from nature. The LVV has a much higher targeting delivery capabilities. And the immunogenicity is also different, because the non-viral has much lower immunogenicity, but the lentiviral has a higher immunogenicity. And the manufacturability is also a little bit different. So here I want to show some challenges for the mRNA and the tLNP for in vivo CAR-T therapy. So typically there are four different aspects for these challenges. Like the scientific level, you have to optimize the delivery specificity to avoid off-target effect, and also the durability and of efficacy. And the second one is a manufacturing process. We want to scale up the samples to give it to as many as possible, as many patients as possible, and The third one is regulatory level. As we know that in China, the government upgrades the policy for the investigation initiating the trial. So there's some change from the government. And also the last one is industrialization. As a kind of on-shelf products, we want to lower down the cost and also bring down the turnaround time as much as possible. So there are a lot of things we need to do to lower down the cost. On the second part, I will introduce ProBio's all-in-one CDMO platform. So as we know, ProBio has the capability to produce the plasmid, and so that's the reason we integrate all these capabilities into our mRNA-tLNP platform. And we can do the plasmid constructions and also the mRNA sequence design and optimizations. And meanwhile, we can do the LNP formulation screening and antibody design and optimization. So all this proof of concept work, it can be done in ProBio. And after we find the best sequence or the good antibody candidate as the binder, we can move to the next step, like the mRNA CMC steps, and also the antibody CMC step. And together, we can make the mRNA and LNP samples. And after we conjugate the antibody to the LNP, we can finally have the TLNP samples. And eventually, we will do a bunch of QC tests. And after all these QC tests, it can be delivered to our customers. So here is a slide that specifically introduced our tLNP platform. As I mentioned, ProBio has the antibody discovery capabilities, so we can handle, actually we have experience in handling different type of antibody format, such as the full-length IgG and VHH FC, and also Fab and single-chain FV, or the VHH nanobody. So all these different type of antibody can conjugate to the LNP and generating the tLNP samples. And then we will use like a TFF to purify, to remove the extra residue or antibody and purify the tLNP samples. And after that, we'll sterilize filtration to generate the final samples, and then fill and finish. So here's some detailed information for our platform. So we can handle different type of antibodies, and also different type of a payload, like the siRNA or the sgRNA, guide RNA, and mRNA. And and this platform is robust and scale up very easily. And so far we have delivered multiple batch of tLNP to our customers. It can be up to gram level, and total mRNA recovery is more than 60%. Oops. So in ProBio, we typically we can use 3 different methods to conjugate the antibody to the LNP, like the surface conjugation, which based on the cell group, and monomer. So it's a covalent binding, and it's a robust process, and a certain limit type of impurity, but it shows a very good consistency. And the concept requires continuous operations, because if you separate these steps, the tLNP, the quality of the tLNP will be affected. So that's the reason usually we would do in a continuous operation way. And the second method is based on the DBCO and Zeta to conjugate the antibody to the LNP. And again, it's a covalent bonding, and it's also a robust process and good consistency. But the cons is it contains some potential of a zeta residue, which is not good for the cell assay or in vivo assay. And the last one is a post-insertion. So for this strategy, basically, we first conjugate the antibody to the mole-pack lipid. So in that way, it will form a small compounds like nanoparticle, small nanoparticle. Meanwhile, we'll also generate a traditional lipid nanoparticle, contain the mRNA payload and also all the lipid components. And then we will mix these two nanoparticles together. It will automatically fusion together and generate the final tLNP products. So the pros is it's very easy for the R&D screening and faster turnaround time. And the cons is because the PDI and will be not so even after fusing these two nanoparticles together, and there are some uncertain impurity. So most of the time, our CMC project will choose the first two methods. And here I will show some case studies. The first one is I will present some data for the tLNP characterization. And you can see this is the picture shows our tLNP generation or the production process. And here on the left below, what we did is we conjugate different amount of antibody to the LNP, and then measure the size and also the PDI for these different samples, and then we try to characterize the encapsulation efficiency, and also try to characterize the samples under the microscope. And you can see when we load different amount of antibody, the size will increase. That is quite easy to understand, because when you add more antibody or conjugate more antibody to the LNP surface, the size will be large, right? And the encapsulation efficiency is quite stable, 96 or 97, and the shape under the microscope is also quite good, very even. And then we try to, as I mentioned, we use the sign group and the monoamide to do the conjugation. So usually we use TCEP to reduce the antibody, to open the sign group between the antibody. And here we test different conditions, either different amount of TCEP, reduced reagent, and also different time point, like TCEP for 2 hours or 4 hours. And as you can see, when we treat the antibody with different conditions, the subgroups between the antibody will be broken in different ratios. So when you use antibody to conjugate LNP, we always recommend to do antibody reducing optimization. So in that way, you will find out the best reduced condition for your antibody. And then after that, we also try to characterize how much mole pack is needed for the conjugation. So in this experiment, we play with different ratio of mole pack and also regular DMG pack. So you can see the total on the right up figure. This is the total pack ratio is 1.5%, and we increase the mole pack ratios, which can be used for linking the antibody from very low level and to almost 1% in the final formulations. And we list as LNP 1, 2, 3, 4, 5. And as you can see, these different LNP samples, they have the size increasing. That is easy to understand, because if you increase the mole pack in the formulation, which means that you will have more antibody conjugate to the LNP, right? And the encapsulation and the zeta potential is quite stable. So after we characterize the best ratio of the mole pack, and then we try to check the gene expression. So we construct different LNP and the TLNP samples as a kind of like comparison. So LNP1 and the TLNP1, LNP2, TLNP2, etc. So, and then as you can see, the gene expression here, we use the GFP as a reporter genes, and we check the GFP fluorescence signal in the experiment. And you can see for the second conditions, TLNP2 has the highest GFP expression, which means the mole pack ratio of this condition is the best one. So after we finalize the mole pack ratio in the formulation, and then we try to characterize how much antibody is needed for this formulation. So we fix the mole pack in this LNP2 formulation, and then add a different amount of antibody, and then try to do the experiment again. So here is one fold of antibody, and 2-fold, 3-fold, 4-fold, and 5 and 6. So we increase the antibody in the formulation, and as you can see, as we increase the antibody, the size increasing, and also the PDI is quite stable. And eventually we try to see the GFP expression for these different samples, and you can see that when we add 3 times of antibody in this formulation, the GFP expression is the highest. So in that way, we can lock the antibody amount in this formulation. So that is a typical process for the TLNP method development. So you have to optimize your mole pack, and also you have to optimize your amount of antibody, and in that way you can finalize your TLNP formulation. So after we characterize all this mole ratio and antibody ratios in the formulation, and then we check, we develop different QC methods to check the TLNP quantity. So here we use the nanoflow cytometry to check the positive rate of TLNP samples. So as you can see, the TLNP sample has more than 95% positive rate here, and the LNP sample basically has below 1%, 0.6%. And we can also use ELISA or nanoflow cytometry to measure the antibody concentration. So this is like the whole antibody concentration in the TLNP samples. And also we can use the nanoflow to measure like the average number of antibody on the LNP. So we can use different methods to characterize the quantity or the antibody conjugated to the TLNP. And after this, we also use the cell assay to validate the TLNP delivery capabilities. So here, we first try the JACA cell assay, and you can see this is the LNP sample as a kind of negative control. Here is the TLNP samples, and this is a lung-specific TLNP, which means, for example, this is like JACA is kind of like a T cell, so we use the CD7 binder here. So that's the reason that TLNP can have much higher protein GLP expression. For this lung-specific TLNP, we use a non-related binder here. And for another groups, we knock out the receptor for the CD7 in the JACA cells. So basically, there's no receptor in these cell lines, and so you can see the TLNP significant drop down. And we also test these samples in the PBMC, and as you can see, for the lung T cells, basically we observe very low GLP expression. But for the T cells, we observe much higher GFP expression, and it's also a dosing-dependent, kind of like a dosing-dependent result. And then we try to characterize the stabilities for these samples, and you can see here I just shows the timeline of 2 months here. We put we use different buffer like the PBS buffer and the trace sucrose buffer to store the TLNP samples, and then we check the size and also the PDI for these 2 different groups. And as you can see, the size and the PDI is quite stable, and also we try to characterize the encapsulation efficiency. It's almost 96 or 97%. And then we try to characterize the freeze and thaw capabilities. So as you can see, we freeze and thaw for 1 time, 2 times, 3 times, and 4 times, and the size actually increase after the 3rd times, and the PDI is quite good, it's acceptable, and the encapsulation efficiency is also quite stable. It's still like 95 or 96% here. So lastly, I want to introduce the ProBio's fill and finish capabilities, and we can do the filling in an automatic way. It will be done in C+A, which was done in an isolator. And then we can do different type of fillings like 2R, 6R, and 15Rs virals. And all these reagents are disposable filling components. And the speed can be up to 1,500 virus per hour for 2 hours, and then 1,020 virus per hour for the 15RO. And the filling volume is 0.2 milliliter to 15 milliliter, depends on the project's requirement. And lastly, I want to introduce our in-house QC capabilities. And as I mentioned, the mRNA and also the TLNP has much more complicated quantities. So we developed different methods to check all these to check the samples. So for example, the LNP, we will check the shape using the CRIN EM, and also the antibody positive rate using the nano flow cytometry, and empty LNP rate also can be checked using the nanoflow cytometry, and the size of PDI zeta potential, which will also be measured by the DIS or ELS. And for the purity, we will check the mRNA integrity and the purity and encapsulation efficiency and mRNA contact, etc. So you can check more details. I will share the slide later, so you can check more details in my PPT later. So summary is like in ProBio, we can provide all-in-one service from the plasmid to mRNA to tLNP for our customers, and our platform shows excellent targeting and expression data, and we have developed multiple QC methods to check the quality of the tLNP samples, and we also have long-term stability data. Today I just showed 2 months. Actually, our data has almost reached the 4 months. And for the CMC scanning up, we can do up to gram level. So, lastly, as I showed in the slide, we have done multiple projects with our customers, and most of them are for in vivo CAR-T gene therapy. So that's all the information I want to share today. If you have any question, I will be very happy to answer.